From a structural point of view, these retroviruses are certainly a homogeneous group. But if you look carefully, you can see, well, there are minute differences in fine structure, and these can be related to the host of a particular retrovirus. So if you think about the equine infectious anemia virus, you know immediately that cannot be a C-type oncovirus. That does not cause, that equine infectious anemia virus does not cause cancer in mice, for example. So we can correlate structure and function to some extent.
And what kind of retroviruses is HIV?
It's
okay.
HIV is definitely a lentivirus, and well, we saw that very early in '84 when we got the cultures from Bob Gallo, and it was immediately clear that cannot be a C-type particle, and that was helpful in those times because nobody knew in '84 and '85 what is a virus, where does it belong. And if you can make a statement, it's a lentivirus, you can think about the function of that virus. Lentiviruses are immunodeficiency viruses. They are causing immunodeficiency in horses, in goats, in sheep, and so on. So making that statement, this is a lentivirus, was important in those times. We did that. In the same year, there was a classification coming from nucleic acid structural analysis. So electron microscopy was very early in the front, in the classification of these viruses.
Now I noticed you made a comparison between lentiviruses and I think you said C-type?
C-type viruses. These are the oncogenic RNA viruses that you find in small laboratory animals, in chickens, in a variety of animals. These can be endogenous viruses. They belong to the genome and are expressed only under certain circumstances in cell culture and so on.
Okay.
Or some of them are spread horizontally as an infectious entity, infectious disease, killing, really killing very acutely chickens, for example. The reticuloendotheliosis virus of chicken is a deadly, immediately killing disease. But the lentiviruses, they need their time because they have to interact with the immune system. And that takes, think about HIV, 10 years. Nowadays, we can interfere with that process. I think that was our first paper on that problem in '85. There was a whole story. HTLV-III, quite different from the lentiviruses, which are shown here. It's a publication, it's very rough stuff in a print, but I can show you later. These are mammalian D-type and B-type viruses causing memory tumor cancer and so on. And here you have the whole story, the structural analysis, C-type and so on. I will give you one of those prints later.
Okay.
You really need to have the good images.
Using the electron microscope or electron micrograph, how easy is it for you to differentiate between all these retroviruses? Because they all look, to someone like me, the untrained eye, they all look the same.
Certainly not. I will be able to teach you within half an hour. We have made a nice summary on these structural differences. You can measure. And if you start measuring the ribonucleoprotein, that's a complex of the genome and some protein, where is it in the virion, in the budding, in the immature particle? How is that organized in the mature particle versus the immature particle? What about the glycoprotein knobs? These are questions that can be quantitatively assessed. So you really can make an objective diagnosis.
So for someone like you, it's easy to tell the difference?
Yeah.
Okay. Now, can you show me a picture of an HIV particle?
That's one of the best things ever published. That is a nice story on the shedding of the glycoprotein. The immature particle, you see there's marker. We have been looking by immunoelectron microscopy for the surface glycoprotein of HIV. Mm-hmm. It's there. These are immature particles. They are budding from the cells. And these are mature particles. There's much more glycoprotein than on the mature HIV. So that's kind of shedding.
So once the virus buds off of the cell,
Yeah.
it sheds its glycoprotein.
Yeah.
It sheds its
Yeah.
glycoproteins.
And you see it's probably a time-dependent process. You see some of them still have a lot of glycoprotein, and some are almost empty. But another thing is interesting. They look different inside.
How come?
How come? Because this is thin section electron microscopy. If you have a cone in this Ebene, in this direction, and you cut it transversely, you end up with such a structure.
So these are the
These
same.
are randomly orientated, and these are very thin slices,
Okay.
and therefore you have different projections, randomly orientation. And you also find some remnants of the envelope. You see the glycoprotein cut very much tangentially here. Okay, these are typical cores of the lentivirus, a lentivirus core. Typical for HIV and SIV are the lateral bodies. These are probably remnants of the internal proteins of the virion that are not used for the assembly. There's probably no function with those things. But here you have the genome inside.
Okay.
But
Do you want these over there?
What do you consider to be the main morphological characteristic of HIV?
it's a cone-shaped core with a mature particle. Mature particle means a cell-released particle after cutting down the GAG protein. In the immature particle, there you see the GAG protein. It's still intact and directly underneath the lipid envelope, directly underneath. Then you have the glycoprotein knobs. They are about 10 nanometer in length. Yeah, they are trimers that we have described 15 years ago. Nobody was believing in the trimeric structure of the GP120. Everybody was going for the monomeric vaccine, which certainly was not correct, because if you have a trimer structure, the presentation of the antigens to the immune system is different compared with a monomeric structure. That was important in those times. I got grilled for a certain while, but now people have come to the trimer structure. Yeah.
Once the immature particle turns into a mature particle, how long does it take before it sheds all of the knobs?
Shedding of the knobs is certainly time dependent. It will need more than 24 hours for shedding of half of them.
So 48 hours and all the knobs are gone.
Yeah, it's statistics.
Huh. Now, what is the size? How big is HIV? What is the diameter?
120 to 150 nanometer.
does that change with its virulence, or is it always the
It's
same
not,
size?
it's not clear whether it really changes with virulence. I think it's well, a question of orientation. Think about the slicing problem. If you have a tomato, you make 3 slices, you are not sure about the real diameter of the tomato. Yeah.
Why is the electron microscope so important?
Because we easily, if the business is done in a correct way, we easily get an idea about functional aspects, structure and function. These are correlated. If you see the virus is losing the glycoprotein knobs, well, you end up with shedding. And then you think about this might have an impact on pathogenesis. If you see these are trimers, the glycoprotein knobs, you should think about the correct formulation of a vaccine. These knobs are the glycoprotein, GP120 trimers, and these knobs are binding to the CD4 on the host cell. And if there are 2 or 3 or 4 bonds closed already, then a conformational change will take place at GP41 underneath, will make the contact with the membrane, and then the whole machinery will work. The things Ron Montelaro, for example, in Pittsburgh has described so nicely.
How did electron microscopy change science, or change the science of HIV?
Well, I think it started to make virology a very basic natural science, piece of natural science. Started in, well, the first virus shown ever by electron microscopy was a pox virus in 38. With that, I think we were able to make a few points. The trimeric nature of the glycoproteins was important. The shedding of the glycoprotein was important. And we also thought about, well, how does that virus enter a cell? There are 2 mechanisms. It can enter via that interaction, GP120, GP41, to the cell, fusion with the host cell. But there's also a receptor-mediated uptake. Receptor-mediated uptake means that this virus will be engulfed into the cell via clathrin-coated pits. And
If the virus sheds the GP120
Yeah.
within 48 hours,
yeah,
how does it infect the CD4 cell?
a good question. Think about the magnitude of virus production. If I get infected, within a week, I will have a burst of virus up to 10 to the 7 particles per ml. 10 to the 7, that means 10 million. 10 million of particles per milliliter of blood. And there's a statistical distribution. A lot of virus will decay, will lose infectivity, but there's also production again and again. So it's a high producer system, and it's just bad chance. Some viruses will be infective. 10,000 will be inactivated already.
Okay. Okay, I see. So it's all about the statistics and
Yeah,
the numbers
yeah,
and
yeah,
probability.
Yeah, yeah.
Okay. Let's go back to the beginning when there was only AIDS and no HIV, just to search for the causative agent by Gallo and Montagnier.
Mm-hmm.
Now, from what I read, the first thing Montagnier did was, before purifying, was take cells from a person he thought were infected, pass it on in culture,
Yeah.
and then take electron micrographs of the supernamed. Now my question is, why can't one just look at the culture through the EMs and definitely say they found a virus before purification? How come you have to go through the purification step?
I think it's not necessary to go for purification. Probably he was interested for purification to make it biochemically safe. But these viruses are the most labile particles you can imagine. They are destroyed by this. You will have still the reverse transcriptase. And having the reverse transcriptase was all in those times. They had some electron microscopy, sure. But they should have done more electron microscopy with a virus producing cultures, as we did. And then you are on the more safe side. No purification. Why purification? The virus is pure and dangerous and infective in culture. If you wait for purification, it will decay. And the techniques are absolutely drastic. There's osmotic damage and the envelopes are gone, and a very dangerous situation for the virus.
Now, Professor Kurth in the interview said that Montagnier really didn't, at least for him, Montagnier didn't convince people that he had found truly a new virus, because he said when he looked at the EMs that Montagnier showed, it was just one, and it wasn't very clear. What are your thoughts on Montagnier's publication?
This is unfortunate politics of the publishers, today at least. In those times I've seen these publications, stamp-size images. It's a nuisance. It's a nuisance. You do not really see much. And I do not know how good electron microscopy was really done, but they probably should have done a bit more, and then they would have been very safe. And electron microscopy is an art and a science. Art and science, and nobody is able to judge what the electron microscopist is really doing. You have to have experts discussing these things. Sometimes people are far from the microscope. They do not understand structure and function. They like to have a nice image, that's all. They do not understand sometimes, and that's a danger. I could imagine if Montagnier would have pushed the fine structure of HIV a bit more, he would have been more convincing. It was a very peculiar thing. And think about, it was a side track. Initially, it was assumed to be a side track. These were people he was studying that were infected by blood products, hemophiliacs. While in the States, the homophiliacs, the gay population was affected. They had to bring these things together. So also the names initially for these viruses were different.
Oh, right.
Yeah.
Have an HTLV.
Yeah, yeah.
Now, one of the things Dr. Francis told me is when at least CDC purified what they thought might be a new virus, they spun it in the high-feed centrifuge and then took a photograph where retrovirus is banned. Why, from an EM perspective, is it so crucial to take a picture of that banding area?
Of course you can do that. They should have done a bit more on the virus-cell interaction. Then the picture would have been very clear. Francis, what they did, purification, look into gradient banded material, would mean they have mature particles. These are swollen, osmotically damaged, and so on and so on. And there's some detritus at the right density. Dangerous things. If you look into a culture, it's very clear what you are looking at. For standardization, it is important. If you think about preparing a vaccine, okay, that's a far goal. If you think about preparing diagnostic kits, you need the virus as an antigen to prepare ELISAs. for serology to test whether somebody is infected, has developed antibodies. You need a controlled antigen. And to control the antigen, electron microscopy is also important.
At least for you, is it important to photograph where the virus is banned in the gradient?
Yeah, because retrovirology has also some history. These are established techniques. If you go to C-type particles, they can be easily bended and they are stable. And if you think HIV is a new retrovirus, you have to go the same way, just to be acknowledged as a retrovirologist dealing with that new virus in a proper way. It might turn out difficult, but why not?
He also said that there's a lot of junk particles and look-alike retroviruses.
Yeah, but that is a question of you concentrate and you isolate the material, but the isolation is not just separating the detritus, the dirt, from the virus. If you separate by density 1.15 gram per ml, you have a lot of vesicular stuff inside, not related to virus. Detritus, a reaction product of the cell. You have a lot of host cell constituents in that band. That's not too nice.
How do you differentiate between, I mean, with so many different particles, how does one determine precisely which one was the cause of AIDS and which one was look-alike and which one were just fragments and things of that nature?
If you go for ultra thin sections in culture, it's no difficulty to differentiate. If you go for gradient purified material, it can be difficult. You have to make sure this structure is an enveloped structure with the proper size and it contains a cone-shaped core. Then you are fine. Then you know it is a virus. If you just see some envelope structures, you cannot make a sound judgment. You can start with immunodetection of virus antigens and so on. Then you can guess, well, this might be 20% of virus and 80% of dirt. This is a relation, yeah? If you go for purified things.
So then for you, the culture is the only thing that's really important?
For a structural analysis, yeah. On the other hand, you have to go to purify the virus as an antigen for ELISA testing, and that was a very important step. And people learned there's a contamination by cellular constituents, HLA, histocompatibility, transplantation antigens. They are coming from the cell. They are sitting on the virus. They are sitting on the virus. That can make complications. And if you choose a cell without the production of these HLA antigens, this antigen is not on the virion. And then there is no cross-contamination with regard to the viral structure. You have to look for a proper production system. And that can be chosen, that takes time. I know this lab situation with Bob Gallo. Bob Gallo is working not much in electron microscopy, but he has a lot of people doing that job. We had been in close collaboration with him. He got our pictures for his articles. So no doubt, these are HIV particles, lentiviruses, which he is working. You can make the statement, well, it's not Bob Gallo's material, it's Montanier's material. There was some mixing up of the cultures. But think about the speed that was necessary in those times, the speed that was necessary to solve the question, what is going on? Cultures were mixed, and I can tell you from my experience, people are mixing up things. That happens.
I didn't realize. So the pictures that was published in the 1984 Science.
Yeah,
Those weren't from his cultures, those were from yours.
Not from, no. These pictures were made in the States, but the virus probably was Montagnier's virus, because he was looking at the cultures that were sent by Montagnier to Bob Gallo. The cultures were mixed up in those places. But these pictures were not so impressive in those times. They were not much better than Montagnier's initial images.
I see. So
And-
you sent him pictures then?
Yeah. And when we came into the business, I was very gracious. Bob Gallo was responding immediately. He sent his cultures to our place, and we were able to do a decent job.
How many pictures have you published on HIV?
Probably some 50.
And how many have you taken?
Oh, that's a good question. Friday night, I usually took 90 images. But this was just a lot of fun, and we were relaxed in those times, and we had good samples. Okay, we prepared some samples 5 times to get the best results. And that is also an answer why were the first images shown by the Montagnier group not so convincing. You really have to do you have to stay with the work.
What is needed to prove that you in fact have a new virus? Because we had AIDS, but we didn't have HIV yet, or we didn't know about HIV. So going back to, take yourself back to the day when we didn't know what it was. What did you have to do to prove that there was a new virus? And how did you confirm once you found something that was the virus?
You could use electron microscopy. Antibodies in an HIV-infected person, they are labeling that virus in cell culture, identifying. That means it's a tag. I'm reactive against that virus. That could be an answer. I think so.
Mm-hmm.
Yeah.
Can you talk about what happened also back within Montagnier and Gallo?
Well, they were hunting, and they were looking for C-type particles first, because they thought, well, on the French side, leukemia, immunosuppression, and these things are relevant to the C-type part of the retroviruses. And also Bob Gallo, well, he thought about HTLV-III, human T-cell leukemia virus type 1, which is transmitted in Asia and in other places with a certain frequency via breast milk. HTLV-III, clearly different from HTLV-III, but still the name HTLV-III. No, it's a lentivirus, so they stopped the name HTLV-III. They went to HIV, because it's a different story. It's a lentivirus story, not a C-type story. Yeah, okay. The pictures, I will show you some, and you put your finger and
Okay.
what you need to have, because I feel some of them might be good. But here you do not see anything about the details. There's no core to be seen because it's just the surface. But I would say it's probably a virus because of the size. It's very small and it's isometric. A lot of particles have the same size. That's nonsense. Hazeltine. Okay, I will give you the. Here you have the model that we did. Yeah, 86. Immunolabeling of the glycoprotein. The localization of the structure. And we got antibodies from all over the world. That was absolutely essential. Here you see the glycoprotein knobs, the trimers, lateral bodies, and so the nomenclature was also new. Nobody knew about lateral bodies in those times. That is let's start with the start. This is a cell. Is there a virus? There is virus. HIV. So small.
This are all of these
Yeah,
HIV.
yeah. And if you use a small magnification, you really cannot be sure about the nature of these things. You have to blow them a bit up. So it's a very low mag. I don't know. It's probably 10,000. Yeah, it's 10,000. It's a model. Another, taken directly from a patient. There's something going on. Something. Higher make. Okay. And another higher make. It's technically not perfect, but the course.
So these are HIV here.
Yeah.
And what are
yeah.
these?
Think about the tomato.
Oh, so are these HIV too?
Yeah.
Everything's
Probably.
Probably.
Yeah.
But this little one here.
Dirt.
Oh, dirt.
These are the vesicles that can accumulate if you look for purified virus.
Okay.
It's in the supernatant, and you concentrate it
Okay.
if you purify. It's not purification. These are the better ones. Too dark, I should admit. But you see the glycoprotein knobs. That's a regular, our regular type of magnification, times 100,000. But very typical, that core.
Now these ones here,
Yeah.
they don't look the same and they look
Nope.
smaller.
Yep.
Are
Again, think about the tomato. If you make a thin section that is covering only the upper region,
Okay.
it will be smaller.
I see.
Yeah, it will be smaller. So you really need interpretation, but that can be difficult. Okay, these are
So-
more of those.
How much
Yeah.
interpretation is there when looking at EMs?
Well, we thought about this core might be a tubular core. You see it. It is here and there. Sometimes you have an assembly of the core constituents, these little beads, the capsomeres, in a tubular way. But that's not contradictory. An assembly of a virus can follow different symmetries. You can make a regular virus like a rotavirus, a stool virus. You can assemble that virus as a helix, as a tubular structure. So there's some freedom to the structure. You just have to know it can happen. It can happen. But the regular form would be something, a cone.
So when I look at this,
Yeah.
in taking into account the tomato,
Yeah.
this core though looks extremely different than say this core here.
I would say so, yeah. I have no idea how to explain this large triangle. I'm used to that structure, let's say, and make a section in that way, then you end up with this one.
But would you say that's HIV too?
Yeah. Think about 10,000 non-infectious, malformed particles, and one infectious.
Now, what about this one here? This one doesn't look like it has a core at all.
I would assume there is kind of a core. There's some in the center, but I just neglect these things because I cannot make a clear-cut judgment. What about that one? There probably is a core cut in that situation. While this one, centrally located, it might be this situation. The interpretation of ultra-thin sections might need some experience, let's say so.
Okay. And
Yeah.
let's see. Now the one thing I
Yeah,
don't see on these.
we have more.
Oh, okay. It looks like the knobs here look like little knobs everywhere. It-
Yeah, okay. There's a chance that you have some dirt, of course, in the cell culture. And these would be knobs from a particle that is cut tangentially. Here the same. That's my interpretation. So my interpretation. But here are the knobs. Here there might be some dirt. Here, sure. That is dirt from preparation.
Mhm. But don't these look like knobs too, coming off of this big one?
Yeah, you know, there are glycoproteins also on the cell.
Oh, okay.
Yeah. Now we have to differentiate between them. If we would like to go for differentiation, we have to use immunoelectron microscopy to make a tag. It's either the glycoprotein or it's something different. Duplicates. Well, what's new here? You find some budding structures. Budding structures.
Mm.
Budding structures.
Now something like this couldn't be HIV, right? Because it has two nucleus.
Why not? A packaging problem. That happens.
Oh, okay.
But that happens also with herpes viruses. And everybody is believing in herpes being an infectious virus.
Right.
You know?
Okay.
Packaging problems, that's nice. You have the tubular arrangement and the cone. Yeah.
Now this wouldn't be HIV though, right? It's too big.
Yeah, but here is budding. Again, the problem of an ultra-thin section. You do not know how this will be connected to the cell. It should be connected, if you like.
Now all these pictures here,
Yeah,
are these all from culture?
all from culture.
Do you have any from the gradient?
Yeah, there's 80% of dirt.
Oh, 80% of dirt.
Yeah.
Oh,
And therefore I didn't like that, but it was necessary also for us to control, because this house in 85 already established ELISA antigen material for testing people. There was nothing commercially available, but we had to purify. We had to look at the material that was used for the ELISA. 80% dirt. Okay.
okay.
That's the truth.
Do you have any pictures that we can look at from the purified?
It's not
Gradient?
here. It's up. We can have a look. Not so nice pictures, just the small things. Here you see the trimers.
I see. Three.
Triangular, and we did include also influenza virus, glycoprotein, the hemagglutinin, which is a trimer structure.
Okay.
And we compared, and then it was very clear, 15 years ago already. Trimers. Okay, you have that. Okay, and here you have a big story. Budding and so on and so on. So, budding can take place very near to each other. This might end up as a virus with two cores.
And you said that the other pictures upstairs, that 80%
Yeah.
was dirt.
Yeah,
What
yeah.
was the other 20%
Virus.
virus?
Yeah. You find bacteria and fungi and everything. In a culture, it's sometimes a mess.
Was it difficult to figure out which was HIV and
No.
which was garbage?
Looking for the right structure, looking for the core. Then you're fine.
You found out.
Yep.
Is it possible to get HIV by itself without any garbage?
There are certainly cultures which are very clean. Think about that one. Well, these are cellular processes, of course, but these are bound to the cell. And if you wash away the virus, it's relatively clean. So I would assume you can end up with a 60% clean virus preparation.
60%.
But you need control.
Yes.
Yeah.
All about control.
Yeah.
Is the size also important in determining what you're looking at?
Yeah. Think about viruses can be as big as pox viruses, up to 300 nanometer. HIV up to 150. The smallest autonomously growing virus will be a circovirus, just 15
Oh, wow.
nanometer.
So what's the smallest HIV particle that's ever been documented?
I think it's something 120 to 150. No, these are fixed morphological entities.
They don't change.
They don't change.
So that's a huge thing in helping you differentiate whether it's HIV or
Yeah,
not.
yeah. The size of a structure is very important to make the diagnosis. The size is the first thing to look at. And then you look for the fine structure,
Okay.
whether there's a cone-shaped core
Okay.
and glycoprotein and the way it's budding from the cell. Important. And also look for the mature structure.
So if I were looking under the scope, I think the only thing I would be able to tell is the size. I don't
Yeah.
think I could look at the fine structure like you.
But
But if I saw something that was 220, I know that's not HIV.
probably not. Well, the magnification of the instrument might be wrong. And you need evidence. Evidence means that you have to make a repeat of your observations.
Oh, so you take multiple pictures.
Yeah, yeah. And it's easy. If you look for the pictures pinned to the wall over there, it's very clear that you have in one view perhaps 10 particles. And then you see there are some variations, and you think about why is that variation? Well, it's a cone-shaped core. Depending on the orientation, you will get it as a cone or just as a centrally located material.
And I don't know if I asked you this, I'm sorry. The cone-shaped core
Yeah,
is very identifiable. It looks very different than a C-type, is that right?
yeah, absolutely.
Okay, or at least to the trained eye.
No, you will see it.
Oh, okay.
Yeah.
You said the problem with cell cultures.
You see a cell nucleus, a cytoplasm. There's a lot of things. What's that? It's dirt. Mycoplasma.
Okay.
Some people have a lot of mycoplasma, but there's virus. There is virus. That was.
But these could be something else too, correct?
That's not virus. Why? Because of the density. It's different in density. You cannot make a clear-cut judgment what is here, but there's something inside of the particles. At least they have a diameter that is near to each other.
What about this here? There's absolutely nothing in the center.
Okay.
Is that a virus, or would that just be garbage?
Just kind of an enveloped structure. There's a lipid envelope. But you see, these are the particles. At higher magnification, things are clear.
I see.
And that is not a lentivirus, that's a D-type particle. We had been interested from our Tübingen times in the identification of retroviruses all over the time. We did some clinical virology at the same time, but the lentivirus and the C-type particles were the scientific problem.
Oh. How come, in your opinion, did Gallo and Montagnier see C-type instead of lenti when they looked under their electron microscopes?
Because they had it in the brain.
Oh, they saw what they wanted to see.
Yeah, yeah.
Some people are very fortunate that they don't have these side effects. But many hypotheses, it's been so prolonged.